Hi, my name is Alexandre. I am from Brazil and I will present my research about the bacterial microbiome of men with lower urinary tract symptoms. This is our pilot study. We have nothing to disclosure. Until recently, we believed that urinary tract was sterile in normal conditions. The standard culture of urine can identify only aerobic fast-growing species, but this is only 3% of the bacteria. The urinary microbiome has only recently been described, and the association of the urinary microbiome characteristics with some diseases is still being studied. The bladder microbiome has been considered the most significant for lower urinary tract symptoms evaluation. To obtain a representative sample of the bladder microbiome, the urine must be collected by bladder catheterization or suprapubic punction. The bladder is a low biomass environment. We have few bacteria inside the bladder in normal circumstances. So the characterization of the bladder microbiome, especially in male patients, is difficult. Our aim was to conduct a pilot study of a select group of men with lower urinary tract symptoms and correlate their clinical and urodynamic findings with the ability to detect and characterize the bladder microbiome. So we select male patients scheduled to undergo urodynamic studies for lower urinary tract symptoms evaluation, and we collected data about age, PSA, IPSS score, OABV8 score, post-void residue, urodynamic findings, especially bladder outlet obstruction and detrusor overactivity, and prostate volume. We collected a urine sample using bladder catheterization during the urodynamics. The bladder microbiome was detected and evaluated by genetic studies. We included 49 patients, but we demonstrated the bladder microbiome in only 24.5%. This is the same described by Bajik and Coles in a recently published study when they used the same genetic evaluation method. We did not find any correlation between the presence or demonstration of the microbiome with age, PSA, IPSS, or other evaluated characteristics. All of our patients had an IPSS score of 9 or more, having moderate or severe symptoms. Considering the whole group of patients, we find that male patients with lower urinary tract symptoms have low diversity for both when evaluated by Shannon index or by the presence of a dominant genus. Here we have the characteristics of the patients and the microbiome that is described as reads and operational taxonomic units, and we have that there is any correlation between these data, but this is the pilot study only. Here we have the description of the microbiome in graphic representation, and we can see that most of the samples have a dominant genus. Most of our samples have one predominant genus that can be easily viewed in this graphic. A healthy microbiome is supposed to be like this. So our conclusion is that we had a low rate of bacterial identification in urine samples. This is only 24.5%, and we have no association with the other characteristic studies. We have low bacterial diversity in patients with flutes, and we have the need to seek strategies to increase the detection rates of the bladder microbiome in order to be more representative. Thank you.